So let's start with the next chapter from embryology, that is the microsporangium and the male gametophyte. So as we know that androecium is the male reproductive and essential whorl of the flower, also called as the stamen. It has three parts: filaments, connective, and the anther. The anther contains microspores or pollen grains in a sac-like structure or chambers that are called as the microsporangium. Now, microsporangium, in case of angiosperms, the anthers are monothecous, one chamber. This is monothecous anther, or diathecous, where you have two lobes, as in hibiscus. The anther with one lobe consists of two microsporangium or two chambers. Thus, it is bisporangiate. So, monothecous anther is bisporangiate, whereas diathecous anther, it is tetrasporangiate, as each lobe would contain the two chambers. So, it is tetrasporangiate. So, this one having two, whereas this one 2 plus 2, 1, 2, 3, and 4. So, 4 are there tetrasporangiate. Here at the base, you have the filament. These are the anther lobes. And in the center, you have the connective. Now, each anther lobe here, it is showing two chambers, and that is why the anther is known as the tetrasporangiate anther. Now, the microsporangium at the very beginning or at an early stage, it has the sporogenous tissue which is diploid, that is 2n, and this will give rise to microspore mother cell which are again diploid. Now, sporogenous cells, they will divide mitotically several times before functioning as the pollen mother cells, so as to increase the number of functional pollen mother cells. Pollen mother cells संख्या वाढावी, कारण directly if it enters meiosis, so they would be reduced and the number of the pollen mother cells will not increase. So before entering the meiosis, these sporogenous cells, they will divide mitotically, and so they would remain diploid, and also the number of the functioning or functional pollen mother cell would increase. Later on, the anther changes in size, hence pollen sac also increases in size. Now, the functional pollen mother cell, which are diploid, will produce a mass of haploid pollen grains by entering meiosis. So, pollen mother cells would undergo meiosis, which is a reduction division, and because of which there will be the formation of haploid pollen grains. So, PMC or pollen mother grains, there will be 2n, that is diploid, and later on, as it will enter meiosis, they will produce the pollen grains which are haploid or N. Some of them, pollen mother cells, they are non-functionals and they degenerate, providing the nutrition to the developing pollen mother cells. Now, this process of development of pollen grains from functional pollen mother cells is known as the microsporogenesis. So, microsporogenesis is nothing but the formation or development of pollen grains from the functional pollen mother cells or PMCs. The anther here, it is made up of a single lobe or two lobes. Already we have discussed it beforehand. Development of microsporangium. So initially, the anther will have only a large mass of homogeneous tissues. Homogeneous tissues, all the tissues which are similar, they are meristematic in nature. Homogeneous, they are all alike and they are meristematic. Now, some of the meristematic cells below the protoderm here, they would become special and prominent, acting as the archaeosporial cells. Archaeosporial cells, they will divide initially periclinally, forming two layers: primary parietal layer and primary sporogenous layer. The primary parietal layer divides periclinally, anticlinally, forming the wall layers, that is the wall of the anther. Whereas the primary sporogenous cells, they would function directly as the spore mother cells, or they would divide mitotically, increasing in number, and then they function as the spore mother cells. The spore mother cells undergo meiotic division, giving rise to four microspores arranged tetrahedrally. So each pollen mother cell or spore mother cell would give rise to four microspores which are arranged tetrahedrally. Structure of tetrasporangiate anther. So this tetrasporangiate anther, if it is TS is taken or section is taken, transverse section is taken, what you observe is the epidermis, endothecium, middle layer, and the tapetum, and the sporogenous tissue. So from above, first will be the epidermis, which is the outermost layer made up of single layer of cells which are compactly arranged, and it is present from the early stage, and it is not derived from the primary parietal layer. The cells of epidermis undergo anticlinal division, so the epidermis increases in the surface area to accommodate what is being formed inside. In mature anthers, the cells of epidermis are gracefully stretched and flattened. The function of epidermis is to provide the protection. Below the epidermis, we have the endothecium. Now, endothecium is the second layer of anther wall and is fibrous layer because of the presence of fibrous band. So the another name for this endothecium is the fibrous layer, present just below the epidermis and formed from the primary parietal cells. It also it is also made up of single layer of radially elongated cells. The tangential walls of these cells, they have a characteristic fibrous bands made up of lignin. These help in the dehiscence of the anther. The cells are called as the stomium, where the endothecium is very thin walled, through which the spores will come out. The stomium helps in dehiscence of the anther. The endothecial layer becomes hygroscopic due to the development of fibrous band, and they play an important role in the enthesis, that is the dehiscence of the anther lobe. Endothecium is absent in anthers where dehiscence is through the apical pore because there is no need of this. Middle layer, this is the third layer below the endothecium, made up of 2 to 3 layers of thin-walled parenchymatous cells. The cells of the middle layer stores the starch grain, and these get crushed at the time of meiosis in the pollen mother cells. So when the pollen mother cells are undergoing meiosis, the cells of middle layer, they will get crushed, which further would provide the nutrition. Innermost, we have the tapetum. Now, tapetum is the innermost layer developing from the primary parietal layer and below the middle layer. It is formed of a single homogeneous layer surrounding the sporogenous tissue. Tapetum cells are large radially elongated, having dense cytoplasm and a prominent nucleus. Tapetum is a nutritive layer for the developing microspores. Below the tapetum, you have the sporogenous tissues. Now, sporogenous tissues are formed from the archesporial initial cells or the archesporium directly. The cells which are formed directly act as the pollen mother cells or spore mother cells, which are diploid in the beginning and undergo meiosis to form the pollen grains, which are haploid. The pollen grains further develop and produce the male gametes. Now, the tapetum, which is present in the Tetrasporangiate anther, there are two different types: glandular or secretory tapetum, and the second one, that is the amoeboid or the periplasmodial or plasmodial tapetum. The first one, glandular or secretory tapetum. As the name suggests, glandular, that which secretes something, and we know that all the glands, they are secreting some secretion. So the name secretory tapetum. Now here the secretory or glandular tapetum will remain intact in its original position throughout the development of microspore. जिथे जागा आहे त्याच जागेवर राहतील, ते जागा सोडणार नाही, तिथनं निघणार नाही. The cells do not degenerate. Instead, it will secrete the protoplasmic content in the form of the plasmodium that surrounds the pollen grains. Secretion will provide the nutrition to the developing pollen grains. Next, we have the amoeboid or plasmodial tapetum. Now, amoeboid or plasmodial tapetum is also known as the invasive or periplasmodial tapetum. Now, here the tapetum will not secrete its content slowly. Instead, the tapetum will break down, the cells will break down due to the hydrolytic enzymes. And whatever protoplasm is present will form a plasmodial mass called as the tapetal periplasmodium. And at once it will come and become associated with the pollen grains until the maturity of the pollen grains. This particular type of the tapetum is the primitive type of the tapetum. Functions of tapetum: as it is the innermost layer, it is the most prominent layer. It provides the nutritive tissue to developing microsporangium, and whatever food material which is passing from outside to the sporogenous tissue, it has to pass through the tapetum. The tapetum also plays an important role in the formation of the pollen wall. There are certain bodies which are known as the Ubich's bodies. Now, these are present in the tapetal cells and they contribute a material which is called as the sporopollenin. Now, the sporopollenin is involved in the formation of the exine of pollen grains. The plastids of the tapetal cell synthesize a material like the pollen kit and tryphin. This material protects the pollen grains from the effects of the ultraviolet rays, and its sticky nature is because of this material, which is helpful in the dispersal of pollens or pollination of pollens by the insects. The enzyme callase is derived from the dictyosomes of the tapetal cells, and these degrade the callus around the pollen tetrads, and hence the pollen grains in a tetrad they get separated. Tapetum is very much essential for the formation of viable pollen grains. Viable means that would germinate and be functional afterwards. The proteins present in the exine wall, they are essential for the recognition of compatible pistils. Compatible pistil जेव्हा पॉलिनेशन होतं तर कुठलाही पोलन ग्रेन येऊन त्या स्टिग्मावर पडला तो जर्मिनेट होईल असं नाही. So if it is compatible, then it will germinate. So this compatibility is determined by the proteins which are present in the exine wall. आणि एक्झाइन वॉल फॉर्म करायला कोण मदत करतं? तर टपेटम. So compatibility कोण determine करते indirectly? That is the tapetal cells. Now the definition of microsporogenesis already we have seen. The pollen grains develop from the meiotic division of the pollen mother cells, and these may be of two different types. First is successive type and second is the simultaneous type. Successive म्हणजे काय? ते एका नंतर एक. Whereas simultaneous एकदम एका वेळेला होईल ते. सक्सेसिव्ह म्हणजे जसं एखादा राजा आहे तो वारतो मग त्याचा मुलगा तो राजा होतो. दॅट इज सक्सेशन म्हणजे एक जातो आणि दुसरा येतो. सायमल्टेनियसली म्हणजे एकाच वेळेला दोन्ही पण. So in successive type of cytokinesis, the partition wall formation takes place in two steps. आता हा एक pollen mother cell. When it will divide in the beginning, it will form the two cells. A wall layer will be there. So what we are getting is a dyad, and this dyad again will divide, and a wall layer is formed at right angle to the first one, and then we'll get the 4 pollen grains. So this formation of wall layer is successive, that is one after the another. Whereas in simultaneous type, when the pollen mother cell it divides by meiosis 1 and meiosis 2, it forms the 4 haploid nuclei, and that is lying in the same cytoplasm. All the 4 nuclei will occupy the 4 corners of the cell, followed by the formation of the partition wall or the furrowing of the mother cell wall in between these nuclei. So we have 4 of this. There is the furrowing. So wall layers which are formed, they are formed at the same time. So that is called as the simultaneous type. Types of microspore tetrad. We are following microspore tetrad: tetrahedral, isobilateral, linear tetrad, T-shaped tetrad, and decussate tetrad. In tetrahedral tetrad, the spherical microspore mother cell divides into 4 microspores by simultaneous cell wall formation into 4 vertical planes at right angle to each other. So what we will get? 1, 2, 3, and fourth one will be on the back side. Isobilateral. Here, figure असेल तर आपण figures बघूया. Okay, tetrahedral बघा. 1, 2, 3, 4 on that side. Isobilateral 1, 2, 3, 4. So in 2 planes they are placed. Decussate. So the arrangement is very much similar to the decussate leaves. Decussate मध्ये काय होतं की 2 are there and other 2 they are at the right angle to the first 2. So these 2 and these 2 they are exactly at right angle. Then linear, which are arranged in a single line, whereas T-shaped 1, 2 and 2 above, they are resembling the capital T letter of the English alphabet. You stop here, continue afterwards.